Alright, welcome back. This is the second part of the meiosis talk, and so we're going to actually talk about the second part of meiosis. Now, in the first part, we referred to meiosis I, which is the same as mitosis in that you have prophase, metaphase, anaphase, and telophase. Now, from meiosis I, before we get into meiosis II, there were some important changes that we can compare against mitosis. So, prophase is similar. You get the dissolution of the membrane, you get the movement of the centrioles to north and south pole, and you get the appearance of the chromosomes with their sister chromatid pairs. So anaphase and telophase are also similar to mitosis. Now, however, here's the thing: the metaphase is different. Now, remember, the difference is actually quite simple. It differed from mitosis in that we draw the centrioles there, and this is the chromosome 1. Now remember, there's two copies of chromosome 1. One came from your mother and one came from your father. So these are cells dividing in you to make sperm if you're male, or eggs if you're female. So chromosome 1, one of them you got from your mother, one of them you got from your father. They line up, as I said, like twins holding hands waiting to, walk down whatever. And so they're in their homologous pairs. In mitosis, those two chromosomes would line up like that on the metaphase plate, and therefore the sister chromatids would get separated when the kinetochore is shortened. That doesn't happen in meiosis I. You keep the sister chromatids together because that whole chromosome goes north and that whole chromosome goes south. So the homologous pairs are separated. So it's different because one, they line up in homologous pairs, which in mitosis they don't. Secondly, when they're lined up like that, you can get crossover between these areas that come close together. So crossing over, and we'll put into that synapsis, because the synapsis is the actual touching, and then the crossing over is the switching. So synapsis followed by crossing over is perfectly valid in that case. So that's two things in metaphase are different. And three, independent assortment. Now that is the understanding that if I redraw this, and use green for male, orange for female, we don't know how, if this was just an 8 chromosome total cell with 4 homologous pairs, we can't, the independent assortment says we don't know which ones are going to go on the top line and which ones are going to associate on the bottom line. So in this case, Chromosome 1 is from dad, chromosome 2 and 3 is from mum, and chromosome 4 is from dad in this cell. We have the opposite, of course, here, because you've got 1 and 4 from mum and 2 and 3 from dad. Now, they could just as equally line up like that. This is independent assortment. We just don't know which of those 4 chromosomes is going to be from, you know, mum or dad lying on the top shelf, and the same is true for the bottom shelf. So these are the 3 things. They line up in homologous pairs, which I'm showing here, the centrioles top and bottom. They can cross over, so they can swap between, and they're independently assorted. So these things together lead to trillions of possible variations by themselves. There's a last piece of the puzzle which isn't involved in metaphase itself, but is worth remembering that of the millions of sperm that are produced and the hundreds and thousands of eggs that are produced, we don't know which one of those millions of sperm and which one of those 100,000 of eggs is going to come together. So that's an even bigger layer, and that takes the total possibility of having your identical twin somewhere else in the world is about 64 trillion, something like that. Anyway, it's a very large number. It's very unlikely to happen. So this is where we got to in meiosis 1. We got to the end of meiosis 1, and we half the chromosome number. So in humans, we went from 46 to 23. We still maintain chromosomes 1 to 23. Otherwise, the egg or the sperm would not be viable. So in meiosis 1, metaphase is the different phase. They line up in homologous pairs, which leads to halving of the chromosome number, 46 to 23. During this lining up, they can cross over and swap gene sites, and they're also independently assorted. So we don't know which parts that you got from your mother or father end up in one cell or the other. Okay, so that's meiosis 1. Anaphase, they still separate out the same way, but this time it's the whole chromosome, not the sister chromatid that's been ripped off. So again, it's similar, not exact. And then telophase, again, we separate the nuclear material out into 2 new nuclei, and then you get cytokinesis giving you the 2 cells. But that's the end of the end of meiosis 1. So if you're asked to compare and contrast mitosis with meiosis, well, you've got variation in meiosis, you've got genetically identical in mitosis, you've got all of these things that you want to be talking about, these 4 points here that I've got on the board, 1, 2, 3, and 4. And we haven't even got to meiosis 2 yet, so that's another one. There's 2 divisions, not 1. Alright. You can probably hear my dog barking in the background. She's found something to get excited about. Alright, so meiosis 2 is the second nuclear division. So now we're starting with a cell that still has chromosomes in the form of sister chromatids. So there's its nucleus. So meiosis 2 goes through the same 4, but this time it's prophase 2, metaphase 2, anaphase 2, and telophase 2. So second nuclear division, we've already halved the number, so if I'd drawn all these out, now it's 23 chromosomes in there, not 46. So what's left to do? Well, prophase, we get rid of the membrane again. Centrioles go north and south, so we're seeing the same thing happen. And guess what? We've halved the chromosome number. I'm not going to draw them all out, of course, and I'm just going to show you that these are now may have been crossed over. So you've got slight, genes from other chromosomes from meiosis I. But look, they've lined up on the metaphase plate here in a line, just like in mitosis. And what you'll find is in meiosis II is mitosis. It just does exactly the same thing. It just does it with half the chromosome number, because that's what meiosis I was mainly there to do, as well as to create variation through synapsis and crossing over and independent assortment. So you get your kinetochores coming out, and there's your metaphase. Looks exactly like mitosis. So mark it in your head that when you're describing meiosis II, if you've understood mitosis, then you're good. You've already learned meiosis II. This is why I go back over meiosis I first. Alright, anaphase. Can't remember exactly how I drew them. Now we get the separation, the kinetochores are pulling toward the poles, and now we've separated the sister chromatids, just like in mitosis. So they're now moving toward the poles, and in telophase, we'll get the redevelopment of the nuclear membranes around the chromosomes. I'm inaccurately including the centrioles in that. They don't go into the membrane, but ignore my bad drawing. And then you get the cell dividing through cytokinesis. Now, you've developed 2 cells in meiosis II, so this ends up producing another 2, so you end up with a total of 4 cells from 2 nuclear divisions. Meiosis II, a carbon copy of mitosis. Remember, in meiosis I, a lot of change at metaphase, which gives the variation and halves the chromosome number. Alright, thank you for listening.